The final slides that I want to cover here relate to a very important concept in experimental design, that is the experimental unit. The definition for an experimental unit is the unit to which the treatment is applied. We use a lot of different synonyms for experimental units. In agronomy, we often refer to plots. Horticulture, often plants will be grown in pots. Animal science, that may be an individual animal or an individual plant. And human research often refer to cases. All synonyms for our experimental unit. The application of a treatment to the experimental unit is, by virtue of the level at which the treatment to which it's being applied, is independent from adjacent experimental units. Replications are experimental units that have been treated identically and, importantly, independently. The design of an experiment requires the allocation of treatments at random among the experimental units. And in the case of a completely randomized design, the allocation of treatments is completely at random. In cases that we'll see in later weeks, there may be restriction in the randomization, and this will require modified experimental designs. In many cases, the experimental unit is fairly obvious, the pots or the plots. But in some cases, experimental units might comprise an aggregation of experimental material. Obviously, a plot of, say, soybeans comprises several rows and maybe hundreds or thousands of individuals within those rows. In animal feeding studies, treatments are often applied to a pen of animals. The feed, say in a feeding study, is applied to the pen rather than each individual animal. So we'll look in some examples in a little bit of different types of experimental units and identification of those. Very importantly, measurements that might be repeated within an experimental unit we refer to as subsamples, not replications. Replications, of course, are experimental units treated identically but independently. Repeated measurements within an experimental unit are not independent in that they are conducted within the same experimental unit to which a particular treatment had been applied. Such repeated measurements must be analyzed as subsamples and not as replications. Let's look at this in a little more detail here. Subsamples. Subsamples are repeated measurements on the same experimental unit. This occurs when the measurements are made at a smaller scale than the experimental unit itself. For example, we may measure the height of 20 plants within a plot. We might get the individual weight of animals within a group that's being fed a specific ration. Or in the case of a grazing study, rotational grazing, when we're rotating from field to field in a rotational manner, observations on fields within that rotational grazing system are subsamples within the experimental unit, that grazing system. Most times we will use the average of subsamples to represent the value for a plot. Although, as we'll learn next week, we can conduct analysis of subsample variation and compare it if we wanted to make a quantitative comparison of the variation within an experimental unit to the variation between experimental units. When we have point measurements, repeated samples That is, subsamples are essential to minimize within treatment or among